Hello and welcome to this introduction to cell biology video. I'm Dr. Aled Roberts, and in this video we're going to look at what factors affect the human microbiota and what effect changes in the microbiota have on our physical and mental well-being. And so it's important to remember that across the human body, each site hosts its own unique community of microorganisms shaped by local conditions. However, an important point about the microbiota is that it isn't static. It changes continually throughout life, from the moment we're born right through to the moment we die. Now, when we're born, we're essentially born sterile with no established microbiota. Then, over the first few years of life, microorganisms begin to colonize the body, and by around the age of 3, we develop an adult-like microbiota. Now, this process is shaped by a combination of environmental and innate factors, where environmental refers to external factors from the world around us and innate refers to internal influences within the body. So let's have a look at some of the key contributors that help shape the microbiota. So first up we have genetics, an innate factor where our genes can shape the internal environment of the body, influencing which microorganisms are able to colonize and thrive. Next we have birth type, which can affect early colonization, as babies delivered vaginally are exposed to their mother's vaginal and gut microbiota, whereas those born by cesarean tend to acquire microorganisms from the skin and the hospital environment instead. Things like feeding type early on in life and the type of food we eat later on in life also makes a huge difference, affecting the overall composition of our gut microbiota, which in turn affects our ability to digest certain foods, but also alters some of the microbial products that may be produced. Next we have infections, which at any point in life can disrupt the preformed microbiota, sometimes allowing opportunistic pathogens to outcompete other commensal microorganisms. And linked to this, we have the immune system, which can affect the overall microbiota as a strong, well-developed immune response helps maintain equilibrium, whilst an immature or weakened immune system can give opportunistic microorganisms the upper hand. From this, we also have vaccines, which help indirectly by preventing infections and therefore supporting the overall stability of the microbiota, and we have antibiotics on the other side, which can dramatically reduce microbial diversity, wiping out both harmful and beneficial bacteria. Therefore, after their use, there is usually a period of recovery as the microbiota tries to rebuild. Now moving on from this, we have some more lifestyle type factors, so hygiene is another important contributor that can cause transient changes in the microbiota. Good hygiene helps prevent disease, but being too sterile can limit early microbial exposure and reduce diversity, which has its own problems. Our interactions with other people also matter. Regular contact with other people, pets, and the wider environment increases microbial exposure, potentially leading to the transfer of exogenous bacteria. Some other lifestyle choices, such as smoking, can alter the microbial composition, particularly in the mouth and respiratory tract, often reducing beneficial species, which has the potential to allow the growth of opportunistic pathogens that may lead to disease. And finally, even things like the season and the weather around us can have an effect and lead to noticeable seasonal shifts in microbiota composition. And so together, these factors interact continuously to shape the complex and ever-changing ecosystem that makes up the human microbiota. Now, a key question we need to ask ourselves is what's better? Is diversity in our microbiota a good thing? We know that some microorganisms are beneficial, forming part of our endogenous microbiota, whilst others can be harmful, acting as opportunistic pathogens under certain conditions. So when we talk about microbial diversity, what does that mean for health? For instance, is it better to have more diversity within our microbiota or less? Well, typically speaking, what we want is a stable microbiota, one where there's a healthy balance between different phyla and where key species are present to carry out essential functions that support the body. And when this balance is maintained, the microbiota works in harmony with us. However, problems can arise when the microbiota becomes unstable. This can happen when there's an imbalance between phyla, when certain species overgrow, or when important species are missing altogether. Therefore, to stay healthy, we need some sort of homeostasis or balance, resulting in a core functional microbiome. Now, when something happens that causes this core functional microbiome to change, we enter a state known as dysbiosis. And we're actually going to look at two different dysbioses now, one that affects physical health and another that affects our mental health. Now, our gut microbiota plays a central role in maintaining gut homeostasis. It helps to metabolize complex materials from our diet into simpler, more useful compounds that the body can actually absorb and use. Now, many of these processes rely on microbial enzymes that we as humans simply don't produce. In other words, without the gut microbiota, we wouldn't be able to extract nearly as much energy or nutrition from the food we eat. And to show just how important this relationship is, some researchers did various experiments to look at how the gut microbiota can impact physical health. And to do this, they used mice and looked at how the microbiota affects weight and metabolism. And so for this experiment, all of the mice were given the same diet and carried out normal levels of exercise. The only difference between groups was their microbiota. And so in so-called normal mice with a healthy and balanced gut microbiota, they were able to maintain a normal weight throughout the study. Then in germ-free mice, which for those of you that don't know were bred under sterile conditions without any gut microbiota, well, they actually lost weight and became noticeably leaner throughout the study, even though they were provided with the same diet. Then the final group was composed of what we might call obese mice, carrying excess weight. Now, they actually had a pre-altered gut microbiota, which resulted in them gaining additional weight even though they were provided with the same diet. And so what this shows is that changes in microbial composition can directly influence energy balance and fat storage. But that wasn't the end of the experiment. They asked some follow-up questions like what would happen if we gave germ-free mice the microbiota from the obese mice. To test this, researchers performed what's known as a fecal microbiome transplant, which is a procedure that's been used in humans and revolves around collecting stool from a donor, which is then mixed with saline and filtered to remove solid particles, leaving behind the microbial community. This microbiota can then be administered to a new host in a few different ways. For example, it can be delivered upwards through a colonoscopy into the large intestine, downwards via a nasogastric tube, or even as freeze-dried capsules that can be swallowed. Now, when the germ-free mice received a transplant from obese mice, despite being kept on the same diet and activity level, they began to gain weight and became obese themselves. This shows that composition of the microbiota can directly influence metabolism and fat storage independent of diet or exercise. So why did this happen? Well, the obese microbiota is skewed. It isn't balanced in the same way that the healthy one is. And so in these mice, researchers found more firmicutes and more actinobacteria, with fewer bacteroidetes. Now, this imbalance means there's a loss of homeostasis, disrupting the normal metabolic functions of the gut, and the result is obesity, which in this case represents a clear example of dysbiosis, a disturbed or altered microbiota linked to disease. But what's really going on here? What are the microorganisms doing to cause such a dramatic change? Well, it was found that the altered microbiota was changing the levels of short-chain fatty acid in the gut. These are small molecules produced when gut bacteria break down complex carbohydrates. Now, the microbiota from obese mice tends to be more efficient at this process, extracting more energy from the same amount of food. Now, the main short-chain fatty acids produced are butyrate, propionate, and acetate, all of which have increased concentrations within the gut of our obese mice as a result of its microbiota. Now, butyrate, for example, serves as the main energy source for colonocytes, the cells lining the colon. When butyrate levels rise, these cells become more active, leading to increased nutrient absorption from the gut. Propionate and acetate have slightly different roles, however, as both are absorbed into the bloodstream, where they can stimulate lipogenesis, which is the process of creating fat, and gluconeogenesis, which is the production of glucose in the liver. And so altogether, these changes promote fat storage and weight gain, helping to explain why an altered microbiota can shift the body towards an obese phenotype even without changing the diet. Now, in addition to dysbiosis of the gut microbiota being linked to obesity, research has started to reveal connections between the gut microbiota and various behavioural disorders such as autism spectrum disorder, or ASD for short. And this is because of a special connection between the gut and the brain, often referred to as the gut-brain axis. Now, this is a sort of connection that involves two-way communication between the gut and the nervous system, and there's growing evidence that disturbances in the gut microbiota can not only influence physical health, as we've just seen, but also our behaviour and mood. Now, for those that don't know, ASD is a broad term covering a range of developmental conditions that typically appear in the first 3 years of life. It affects social interactions, communication, and often involves repetitive behaviours or unusual interests. And so, whilst genetics plays an important role, environmental factors also contribute, and one particularly interesting risk factor is known as maternal immune activation, or MIA. Now, MIA occurs when the mother's immune system becomes activated during pregnancy, for example, following a viral infection, which leads to increased levels of inflammatory molecules within the blood, placenta, and amniotic fluid, which can then influence the developing nervous system of the fetus. Now, just as we saw before, we've used mouse models to explore this interaction. And so mice under standard conditions where there is no maternal immune activation, they were shown to develop good, open field exploration skills and have a good level of vocalization and social interaction with other mice. Now, to contrast this, some mice had maternal immune activation induced during pregnancy, and the resulting offspring demonstrated lower exploration skills and a reduced level of vocalization and social interaction with other mice, two traits associated with ASD. So why did this happen? Well, the MIA mice were found to have a skewed microbiota, meaning it isn't balanced in the same way as the microbiota of healthy mice. And so in these mice, researchers found an increase in Clostridial species and a decrease in bacteroidetes, which resulted in a number of changes. The first change was a loss of intestinal integrity, and the second was a huge increase in the production of 4-ethyl phenyl sulfate, or EPS. This resulted in elevated gut concentrations of EPS and subsequently increased blood concentrations of EPS. Now, this compound is produced by the altered gut microbiota when they metabolize the amino acid tyrosine, and its presence is linked to anxiety-like behavior, suggesting that this microbial product can directly influence brain function. Now, the interesting thing about this experiment is that just as before, the researchers took this a step further. In order to restore a normal gut microbiota, they fed the MIA-induced mice the human commensal bacteria, Bacteroides fragilis, and symptoms in these mice improved. After this treatment, the researchers noted that the mice had reduced anxiety and better communication, which correlated with lower levels of EPS and improved gut barrier integrity. And so from all of this, it was concluded that these beneficial bacteria help displace the 4 EPS-producing microorganisms, restoring the gut's chemistry to a more normal state. Essentially, by rebalancing the microbiota, the researchers were able to alleviate behavioural symptoms. And so this experiment highlights the potential influence of the gut-brain connection and how microbial metabolites could play a role in other conditions that affect the brain. Now, of course, we need to caveat all of this, as this research is still in its infancy, but the results provide a compelling example of how changes in the gut microbiota can influence not only our physical health, but also our behaviour and mental well-being. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.